Sonication by Tori Dean, Fallon Fowler, Michaela Mickle, and Rhea Corder. Sonication uses sound or acoustic energy to create highly energetic microbubbles that produce extreme physical and chemical reactions. These reactions are a form of sonochemistry. Sonication methods became of interest in the 1920s with the work of Alfred Lee Loomis, a physicist in some capacity, and Edmund Newton Harvey, a zoologist. During this time period, the destruction of microbial cells was of importance in research. Therefore, sonication was heavily researched. Their research focused on investigating the effects of sonication on a rod-shaped marine bacterial species known as Bacillus fisheri, which has bioluminescent properties. The goal was to examine how sonication would affect these bioluminescent properties, specifically how it would reduce them. They concluded sonication had a significant impact on bacterial cells. In the 1960s, the goal was to understand exactly how sonication affected microbial cell interactions. They were studying sonication methods that caused cells to be disrupted or lysed. Some of the physical and chemical processes that were found underlying successful sonication was cavitation, which resulted in rapid heating and cooling rates, free radical generation, and extreme thermal and barometric conditions. In the 1970s, it was found that even brief exposure of bacterial cells to sonication would result in the thinning of the cell wall and the cell wall becoming unattached.
Sonication is most commonly performed using an ultrasonic bath or an ultrasonic probe. The probe is known as a sonicator and is one of the most preferred methods for sonication because of its high intensity range.
Here is a slide showing the advantages and disadvantages of using a probe or a bath. Usually, probes are favored because of their wide intensity range, but often have issues with foaming and the need to be precisely placed in your sample to obtain the best results. Baths are cheaper and can run multiple samples simultaneously, but often are not near as intense as probes.
Here is a short video about how to sonicate. Sonication is a mechanical means of disrupting the cells and separating cellular proteins and lipids. If your technique is not performed correctly, then you may obtain undesirable results. There are different sized probe tips based on particular sonication processes, so it's important to select the proper size. Looking at the chart, you may see that each probe has a recommended sample volume range. Sample volume size and shape is also a factor when considering what tip to use. Using the proper size probe will not only reduce the processing time, but also increase the lifespan of the probe. In general, a small tip will create excellent cavitation effects and easily disrupt surrounding cells with high intensity. Small volumes, usually no more than 50 milliliters, require small tips to fit inside the sample tube. Because microtips offer high intensity, they are made for short processing times because of the amount of heat they produce. Due to this reasoning, the pulse mode should be used to prevent heat buildup. Larger tips offer the advantage of reaching a greater quantity of solution but will not produce an intense reaction. A narrow vessel is preferable to a wide vessel. The ultrasonic energy is generated from the tip and is directed downward. As a sample is processed, the liquid is pushed down and away in all directions. If the vessel is too wide, it will not mix effectively, and some samples will remain untreated. So what type of tip should you use? If you're working with an aqueous sample, then you should consider using a replaceable tip. Replaceable tip probes have threaded ends, and when the tip is worn out, it can be unscrewed and replaced. So when should you not use a replaceable tip then? The answer is whenever you are sonicating a solution that contains organic solvents, alcohols, or any low surface tension liquid. The liquid will seep into the threaded tip and loosen, causing the sonicator to overload. Solid tip probes can typically be used for any type of sample. Probe tips must be submerged properly. If the tip is not submerged enough, the sample will foam or bubble. If the tip is too deep, it will not circulate the sample effectively. Both conditions will end up with poor results. Foaming often occurs with sample volumes below 1 milliliter. Foaming can also be caused when the amplitude setting is too high. Figure A will create foam, Figure B will not circulate, and Figure C indicates correct setup.
Sonication uses waves of acoustic energy to form cavitational microbubbles. These bubbles are responsible for producing extreme temperatures and pressures that drastically change a solution or sample. These waves are made up of ultrasonic frequencies which agitate, move, or vibrate particles in a medium. It is in this way that ultrasound transfers energy to a solution. Sonication forms high-energy gaseous microbubbles known as cavitational bubbles. When these microbubbles collapse in on themselves, known as implosive collapse, they expel a massive amount of stored sound energy which, in turn, creates extreme conditions. But how does this process occur, and what forces are at play? We'll take a further look at the cavitational process and its 3 stages. Cavitational bubbles are spaces of gases that are surrounded by liquid. The liquid surrounding the microbubble exerts a pressure on it, while the gases within the microbubble exert a pressure back. Therefore, microbubbles form where the pressure of liquid is lessened, such as where prior cavitational bubbles were, or vapor-filled crevices. Cavitational bubbles undergo acoustic cycles, namely those of a compression and expansion. Acoustic cycles are influenced by acoustic energy absorption, which is, in turn, influenced by the surface area of the microbubble. The greater the surface area, the more acoustic energy absorbed, the greater the vapor pressure from within the bubble, and the faster the expansion against the liquid solution. Thus, the microbubble stores acoustic energy as it grows. There is a point when the cavitational bubble can no longer efficiently absorb acoustic energy. It is at this point where the microbubble is overgrown. The positive vapor pressure within the microbubble cannot withstand the negative pressure of the surrounding liquid. The liquid rushes in to implosively collapse the microbubble, while the gases inside rapidly condense and violently collide into one another. The intermolecular collisions happen within microseconds and are known to generate plasma. Plasma is a bundle of highly energetic gases that can reach temperatures up to 20,000 Kelvin and 1,000 atmospheres.
Here is a slow motion video of a hydrodynamic cavitation bubble implosively collapsing. Hydrodynamic and acoustic cavitation bubbles undergo the same process, but acquire their energies from different sources.
Back with another one of those block rocking beats. Back with another one of those block rocking beats. Back with another one of those block rocking beats. Back with another one of those block rocking beats.
Implosive collapse forces all of the energy in the form of shock waves to be expelled at one point in the microbubble. When the gases inside the microbubble rapidly condense, these particles collide with one another, forming cavitational shock waves. These waves create microjets and liquid turbulence, which you can see on the picture in the bottom right, and these heavily reform particle surfaces or help to mix solutions. These waves can also cause particles to aggregate and crash into one another at half the speed of light. This can cause intense melting of the particles' surfaces, but it can also cause particles to disassociate from one another. Either way, one or both of these forces can reduce the thickness of the oxide layer coating all the particles. Once this layer is removed, reactivity of the particle is usually enhanced, and this is known as depassivation. The picture on the top right shows a microbubble collapsing, and it is at this point where the mechanical, thermal, and chemical effects occur. Pyrolysis and combustion both involve the lysis of chemicals due to thermochemical decomposition, but one occurs in the presence of oxygen, combustion, and the other in the absence, pyrolysis. The lysis of chemicals within the microbubble creates highly reactive and energetic free radicals, typically through redox reactions. Secondary reactions occur when these free radicals interact with chemical species outside of the microbubbles. These radicals can decompose or synthesize other chemical species. Mass transport is a type of diffusion of molecules across a boundary, like that of a cell's membrane. Sonication thins this diffusion layer, which allows greater entry and escape of molecules. All of the mechanical and chemical effects of sonication affect the biological outcome of your samples. Here, thermal effects destabilize a cell by fully or partially lysing the cell. Here, the mechanical effects of turbulence and microjets shear and break apart DNA and other sensitive cell organelles and proteins. Here, the chemical effects of producing free radicals further destabilizes a cell because of secondary reactions. Implosive collapse causes stress-induced changes that ultimately cause the rupture of the cell's membrane and allow for the entry and escape of molecules from the cell by enhancing mass transport. Sonication has a variety of applications that it's used for, as listed here, but it mainly offers 5 basic results, such as catalyst activation or deactivation, chemical synthesis or decomposition, mixing of solutions, sample erosion, and cell disruption. These results can be seen in the pictures on the slide. The top shows the mixing of 2 solutions, the one below shows the depletion of microbes, and the one to the right shows the agglomeration or aggregation of particles.
Sonication is currently being used to enhance research in many different fields. Researchers in China are using sonication to affect how low molecular weight gelators self-assemble, which could be used to develop shape-shifting materials. Researchers in Sydney, Australia are using sonication to transform graphite into single-layer graphene sheets, creating a very strong and highly conductive material out of inexpensive graphite. Researchers in Sweden are examining the effects of ultrasound on different types of bacteria that infect prosthetic hip and knee implants. They found that specific frequencies and durations within ultrasonic baths can effectively kill gram-negative bacteria, such as E. coli. Researchers in Melbourne, Australia are studying how to more effectively process dairy in large-scale reactors. They were able to show that continuous sonication of concentrated whey proteins reduced viscosity and protein agglomeration sizes while improving protein stability at high temperatures. While sonication is already used for dairy processing, further applications of this technology are being explored for the food industry. Many researchers are trying to use sonication to tenderize meat. The acoustic tenderization process reduces connective tissue content and enhances protease activity. Preliminary results have shown that acoustic cavitation and the accompanying heat improve the tenderness of meat without changing its color, which is important to consumers. Another important field that could potentially benefit from sonication is plant biotechnology. Many scientists across the globe are studying how cavitation caused by low-frequency ultrasound could enhance growth of edible plants. In addition, it has been shown that ultrasound can enhance efficiency when creating recombinant soybeans. Researchers in Japan have developed a process to remove cells from human organs using sonication, so that the remaining extracellular matrix can be seeded with the new cells for transplantation. While their experiments currently use bovine meniscus tissue samples, their goal is to eventually apply this process to human tissues. Finally, researchers in Italy are developing novel procedures to more effectively extract virgin olive oil by pre-treating olives with high-power acoustic waves before crushing. They believe they will be able to extract more high-quality oil per olive.
Thank you for watching a presentation, and I hope you enjoyed it. If you're interested more in the topic of sonication, please check out some of our references.